Okay, this is the lecture for Chapter 3, which is all about the nervous system. As a reminder of things that are coming up, for today we were supposed to have Quiz 2 in class. That's now a take-home assignment. You should have received information about that in the email. Your Amy paper is still due by 7 o'clock on Moodle today. You should have read Chapters 3 and 4 of the textbook. So this is the lecture for Chapter 3. There's always also a video for Chapter 4. And you should have read Dreamland pages 47 to 85. There's a worksheet for that as well. Next week, we're still having the first exam. We will have a little bit of time before the exam begins to go over questions you may have about the material. And you also need to read Dreamland pages 86 to 131. The following week, February 13th, that's the day we're going to be talking about marijuana. There will be a quiz that day. You should read Chapter 9 from the textbook, as well as pages 132 to 184 from Dreamland, and there will be an article assigned additionally. So, the nervous system is divided into two main branches. The central nervous system, which contains the brain and spinal cord, which you see here in pink, and the peripheral nervous system, which is basically everything else that's still part of the nervous system. So if we look at branches of the nervous system, the central nervous system, again, is divided into the brain and spinal cord. The peripheral nervous system can be divided into the somatic nervous system, which is involved in interacting with the external environment. It has both sensory and motor fibers. So, for example, it's involved in touch, seeing, hearing, and then movement when you actually interact with the external environment. The autonomic nervous system regulates the body's internal environment, and it is composed of two branches, the parasympathetic nervous system and the sympathetic nervous system. So, the autonomic nervous system regulates smooth muscle, cardiac muscle, secretions from glands. The two branches, again, are the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system is involved in stimulating, organizing, and mobilizing energy resources in threatening situations, and so it's associated with psychological arousal. This is basically stress. This is the system that's most activated when you're feeling stressed. The parasympathetic nervous system is involved in conserving energy, and it's more activated when you're feeling psychological relaxation. Sometimes people call it the rest and digest system. So organs are constantly receiving input from both the sympathetic nervous system and parasympathetic nervous system. It's just about which is more active at a given time that determines kind of the pattern of activity and the effects that you see. So part of the effects of the sympathetic nervous system are accomplished by the release in action of neurotransmitters. Neurotransmitters are chemicals that regulate nervous system functioning. I'm going to get into more detail about them later in the lecture, but just to very briefly talk about a few of them to talk about the sympathetic and parasympathetic nervous system. One category of neurotransmitters is catecholamines. This includes epinephrine and norepinephrine. And these are associated with activation of the sympathetic nervous system. So they have effects like increasing heart rate, blood pressure, respiration, perspiration. They also decrease the rate of digestion. So these are things that you associate with psychological stress. Acetylcholine is a different neurotransmitter that's involved in the parasympathetic nervous system. And so some of its effects are going to be to decrease heart rate, blood pressure, and respiration, and to increase digestion. So when you think of the sympathetic nervous system, you should think about stress, increased heart rate, blood pressure, respiration. When you think about the parasympathetic nervous system, you should think about relaxation and decreased heart rate, blood pressure, respiration, and increased digestion. So we're going to leave neurotransmitters now and come back to them later in the lecture and now talk about the brain. So the brain can be divided a number of different ways. You could divide it in just three categories, looking at the hindbrain, the midbrain, and the forebrain. You could further divide the hindbrain into two parts, the myelencephalon and the metencephalon, and the forebrain into two parts, the diencephalon and the telencephalon. We're going to get into details of these now, and we're going to start from the bottom up. So an important thing to know about the brain that helps you to think about what functions are localized where is that the brain evolved from the bottom, or the spinal cord, up. So you're going to have things that are controlled by these lower regions of the brain be things that are more necessary for survival, more basic. Whereas things that are controlled by parts of the brain that are further from the spinal cord, and right here the prefrontal cortex is the last part of the brain to evolve, or the most recent part to evolve, are going to be things that are the most complex, most uniquely human. This is going to be like planning, decision making, whereas down here we have things like controlling heart rate. But now let's get into more detail. So the hindbrain, again, right down here, it's composed of the myelencephalon and the metencephalon. The myelencephalon is also known as the medulla. This is the most posterior division of the brain, meaning it's the lowest down, connects to the spinal cord. It mostly contains tracts that carry signals between the rest of the brain and the body. It also has an area that controls vomiting, which is important when we think about drugs and overdose, such as alcohol. And it contains the reticular formation, which is sometimes also called the reticular activating system. The reticular formation is involved in a lot of basic reflexes, such as heart rate, blood pressure, breathing. It helps to regulate those things. It helps to regulate sleep, attention, movement, and some basic muscle tone. So this diagram is showing the reticular formation here. It actually goes through multiple structures. So you see here, part of it's in the medulla, part of it is here in the pons, which is part of the metencephalon, and then going up into the midbrain or mesencephalon. Because the reticular formation is involved in these very basic reflexes that are important for survival, damage to the reticular formation is life-threatening. Someone can die if their reticular formation is damaged. It also has receptors and is sensitive to opioids, alcohol, and barbiturates. So when people die of overdoses from any of these drugs, it's usually because they stop breathing, and that's because it's affected that part of their reticular formation. So moving to the metencephalon, we have the pons, which we just looked at, that contains the reticular formation. We also have the cerebellum here, the striated or striped portion of the base of the brain. It's involved largely with balance and precision of movement. We're learning more about how it's also involved in some learning reflexes. But this is important to think about with drugs because alcohol affects the cerebellum first. And that's why when people drink, they sometimes stumble around more and have difficulty with coordination. So moving into the midbrain or the mesencephalon right here, you also have the reticular formation, as we saw in a previous slide. There's a number of structures in the midbrain. This diagram here is showing you a slice of the midbrain, highlighting some important structures that I want you to know about. One is the periaqueductal gray, which is this part right here. It surrounds the cerebral aqueduct, which is where cerebrospinal fluid flows. This is an important structure. It has a lot of opioid receptors, and so it's involved in analgesia, or the release of pain, that occurs with opioids. They've actually been able to do surgeries on animals without giving them painkillers, but instead just by electrically stimulating this area of the periaqueductal gray here. The substantia nigra, which you'll see here, it's named so because it's in a healthy brain. It looks kind of black in appearance. It contains a lot of dopamine-releasing neurons that are involved in controlling the body's movements, and it's degeneration of the substantia nigra that you see in Parkinson's disease, which is a movement disorder. Moving up to the diencephalon, which is part of the forebrain, it's top of the brain stem here. This is primarily two structures. The thalamus, which if you look here, these two lobes here represent the thalamus. We consider the thalamus to be involved as a relay station. So it's a relay of sensory impulses as they go to the cerebral cortex. So for example, for your visual system, you have neurons that leave your eye, go to the thalamus, synapse there, and then go on to the primary visual cortex, which is here at the back of the brain. The hypothalamus, which is hanging right below the thalamus right there, it receives input from the cerebral cortex. It does actually a lot of things. It helps to regulate the reticular formation and the pituitary gland. People say it's involved in the 4 Fs: feeding, fighting, fleeing, and fornication. Not the other one. And then finally, getting to the telencephalon, or the cerebrum. This is the largest part of the brain. It's what people generally think of when they think of the brain. It's involved in the most complex functions, such as voluntary movement. It interprets sensory input, so it interprets the things that you see, hear, etc. and it mediates complex cognitive processes. There are a lot of important parts of the telencephalon. I'm just going to highlight a few. So one way we can divide the telencephalon is into 4 lobes, although now there's starting to be a 5th one. The occipital lobe back here is involved primarily with analyzing visual information. The temporal lobe down here is involved in analyzing auditory information. It has some memory functions as well. The parietal lobe up here involves using vision to interact with the environment, but also contains the primary somatosensory cortex, so it's involved in analyzing touch. And then the frontal lobe here is kind of the most recent in evolutionary history, so you're going to have this be involved in the most complex activities. So the primary motor cortex is here, directing movement. You have the prefrontal cortex, planning, decision making, and we'll see a little bit later that we also see differences in the prefrontal cortex for people who have had problems with drug abuse. And then the insula, which is below these, is starting to be talked by some people as a 5th cortex, is involved in kind of mind-body connection and self-awareness. So some other important parts, the cerebral cortex is basically what you see. It's this outer layer that's really wrinkly. The basal ganglia is a set of structures that are connected to each other. They're involved in smooth muscle contractions, so making your muscle movement smooth, as well as overlearned sensory motor pathways, meaning if you know how to drive a stick shift, you don't think about it anymore, you just do it. If you know how to shoot a basket, you don't think about all of the specific movements you do. You've learned it, and it just takes over. The amygdala, which I believe there's a picture of on the next slide, is involved in emotion, and it's particularly, we know there's a lot involved in fear. Now, whether it's more involved with fear than other emotions, or fear is just easier to study in animal models, it's hard to say. And then the hippocampus, which is involved in memory formations, the use of spatial memory, and seems to be involved in mood as well. So the limbic system is a set of interrelated structures that are involved in learning and memory, as well as emotion and motivation. And again, here, if we cut away part of the brain, you can see the amygdala right here and the hippocampus here. The hippocampus, again, is involved in memory and potentially mood, and the amygdala here is involved in emotions and fear. I think we can all agree that our brain and spinal cord are important to us, and so there are a number of ways in which they are protected. They're encased in bone, so the skull and the vertebrae. They also have the blood-brain barrier. This is important, particularly when we think about drugs. If we look at capillaries, which are the smallest blood vessels, if you look at capillaries in the rest of the body, the cells that create the vessel walls have kind of gaps between them that allow the flow of substances into and out of the blood, like nutrients, waste, etc. In the brain, they're much more tightly packed, and this serves to keep most toxic substances out, but not necessarily all toxic substances. And so any drug that has a psychoactive effect, by definition, it or one of its metabolites is able to cross the blood-brain barrier. Otherwise, it wouldn't have an effect on thoughts, feelings, behaviors. Drugs that are more fat-soluble are going to cross the blood-brain barrier more easily. There's also 3 meninges, and so these are membranes that surround the brain, between the brain and the skull. You can see them right here. And cerebrospinal fluid, which supports and cushions the brain. So, moving now to smaller structures within the brain. The brain is composed really of neurons and glial cells. We're not going to focus on glial cells at this point in time. We're just going to focus on neurons. So these are the cells of the nervous system that we know the most about and seem to be the kind of important cells of the nervous system. Some of the basic parts that you should know, you have the soma or cell body right here, and that contains the nucleus and some other necessary structure. The nucleus is where you'll find the DNA and chromosomes in a cell. You have the dendrites here projecting off of the soma, and the dendrites receive messages from other neurons. The axon is this long projection here. It carries information from the cell body to the terminal buttons in the form of an action potential, which we'll get to in a second. And it may or may not be covered in myelin sheath, which is shown in blue here. In reality, myelin's a white fatty substance. Myelin serves to increase the speed of conduction, so it increases the speed by which the message would travel down to the terminal buttons. And the terminal buttons here at the end release neurotransmitters, which we'll get to later. And so this allows them to send messages to other neurons. So we can divide neurons broadly into 3 categories, really, but I'll talk about 2 primarily. Sensory neurons are neurons that are carrying information toward the central nervous system. And motor neurons are neurons that are carrying information away from the central nervous system. You can also have interneurons, which are involved in kind of communication between neurons. So again, here's another diagram of a neuron to look at. Here's your somer cell body. There's your nucleus. Inside there, you're going to find DNA. Here are dendrites where this neuron can receive information from other dendrites. At the axon hillock here is where an action potential can start, which is an electrical impulse that allows information to be transmitted between neurons. The electrical impulse, the action potential, would travel along this axon here. This axon is myelinated, so it would jump from one node of Ranvier, which is the little space in between the myelin, to the next. It would be faster than if it wasn't myelinated. And then it would reach the terminal buttons here, where a neurotransmitter would be released to then communicate with the dendrite on this neuron and have an effect on influencing whether or not this neuron is going to fire an action potential. And so the gap here between the terminal button and the dendrite is called a synapse. So it's not something you can actually touch, it's just a gap between adjacent neurons, and that's where communication from one neuron to the next occurs. So at rest, meaning when an action potential is not occurring, the inside of a cell, specifically a neuron we're talking about here, is more negative in charge than the outside of the cell. That's because of the presence of different ions. These are electrically charged particles that are in different concentrations inside versus outside of the cell. And the cell has a membrane that separates the inside of and the outside of the cell that's selectively permeable, meaning that it allows some things to cross but not others. And so at rest, you have a lot of negatively charged protein ions inside the cell, which make it more negative inside the cell relative to outside of the cell. You have a higher concentration of potassium inside the cell than outside the cell, and outside the cell you have higher concentrations of sodium and chloride than you do inside the cell, although all of these are going to exist both inside and outside the cell. It's just about relative concentrations. So, action potentials are rapid electrical signals that travel down an axon. They begin at the axon hillock, which is the junction between the cell body and the axon, and then travel down the axon. I have a picture here of someone touching a hot stove to remind you what the point of an action potential is. If you touch a hot stove, the message that tells your brain and actually spinal cord that it was hot is transmitted by action potentials. There are neurons that are responding to that, send an action potential to tell your brain that it was hot. It actually first tells your spinal cord, and it's a reflex where you pull your hand away because it synapses on an interneuron that synapses on a motor neuron to control muscle contraction. And you pull your hand away before you even realize that it's hot. If you think about when you touch something hot, you'll jerk away before you even process the pain. So, an action potential is this electrical signal, travels down the axon. When it reaches the terminal button, neurotransmitters are released. So at rest, the inside of the cell is more negative than the outside of the cell. We say it's around negative 70 millivolts. When a threshold of excitation is reached, and so that means that you have less of a difference between inside and outside of the cell, an action potential then occurs. When that threshold of excitation is reached, there are sodium channels that are voltage gated, meaning they respond to this change in voltage, and they open. Because there's more sodium outside the cell than inside the cell, and because it's a positively charged ion that's attracted to the negative charge inside the cell, When the sodium channel is open, sodium rushes into the cell. You then have more potassium channels open, and potassium is going to rush out of the cell because it's going to go to an area with less of a potassium concentration. So if we look at a diagram of an action potential, this is what an action potential looks like when we graph it. On the x-axis is time, on the y-axis is membrane potential, which is the difference in voltage between the inside and the outside of the cell. So here at rest, we're looking at about negative 70 millivolts, so it's more negative inside than outside the cell. Because of communication from other neurons, there's a change in this, and the threshold of excitation, which is right here, is reached. When this happens, a bunch of sodium channels open and sodium rushes into the cell. This change in membrane potential also causes potassium channels to open and potassium begins to leave the cell. Then the sodium channels close. They become refractory, so sodium can no longer enter the cell. Potassium channels take longer to close. And eventually, resting potential is reestablished, but only after overshooting and hyperpolarizing the membrane. And so this is showing what's happening in the channels, basically. So again, you're reaching the threshold of excitation. The sodium channels are opening here as the membrane potential changes so that you're getting closer to being more positive inside the cell. Then potassium channels open and potassium exits the cell. The sodium channels then become inactivated or refractory here, so they close, and at first they're at a point where they cannot open again. Potassium's continuing to exit the cell, and then down around here, sodium channels become deactivated or reset, so they're still closed, but they're in a form at which they could actually open again, and then potassium channels close. So, a brief Simpsons clip for you here.
Oh, she's here. Cover for me.
One fugu.
Not fugu. If it is cut improperly, it's...
Yes, it is poisonous, potentially fatal, but if sliced properly, it can be quite tasty.
I must get the master. Oh, Miss Crabapple. Oh, your hair smells so clean. Master, you are needed in the kitchen.
I said cover for me, damn it!
But Master, we need your skilled hands.
My skilled hands are busy. You do it!
Poison, tasty fish. Concentrate.
I want fugu! Fan-fugu-tastic. Beautiful language, isn't it, Mark?
God's sake, don't eat another bite.
Oh, I couldn't possibly.
Mr. Simpson-san, I shall be blunt. We have reason to believe you have eaten poison.
Poison? What should I do? What should I do? Tell me quick.
Oh, no need to panic. There's a map to the hospital on the back of the menu.
Try something new, Homer. What'll it hurt you, Homer? I never heard of a poisoned pork chop.
Your wife agreed that I should break this to you.
No need, Doc. I can read Marge like a book. Ooh, it's good news, isn't it?
No, Mr. Simpson.
Oh.
If, in fact, you've consumed the venom of the blowfish, and from what the chef has told me, it's quite probable, you have 24 hours to live.
24 hours?
Well, 22. I'm sorry I kept you waiting so long.
Oh, Marge, I'm gonna die. I'm gonna die.
Well, if there's one consolation, it's that you'll feel no pain at all until sometime tomorrow evening when your heart suddenly explodes. Now, a little death anxiety is normal. You can expect to go through 5 stages. The first is denial.
No way, because I'm not dying.
Second is anger.
Why, you little duck!
After that comes fear.
What's after fear? What's after fear?
Bargaining.
Doc, you gotta get me out of this. I'll make it worth your while.
Finally, acceptance.
Well, we all gotta go sometime.
Mr. Simpson, your progress astounds me. I should leave you two alone. Perhaps this pamphlet will be helpful.
So you're going to die.
Okay. I assure you that this is in fact relevant. In the blowfish, there's a venom called tetrodotoxin, which blocks sodium channels. If you block sodium channels, that means no action potentials can occur. And so your heart doesn't explode, but what happens is you would become paralyzed, including your hearts and lungs. And so it can kill you if your heart can no longer contract and you can no longer breathe. So they are important because they're involved in everything that you do. So the action potential travels down the axon, and when it reaches the terminal buttons, this is a close-up of a terminal button here and a synapse. A synaptic vesicle that contains neurotransmitters fuses with the membrane. The neurotransmitters are released into the synapse and then bind with receptors on the postsynaptic membrane. So here's your terminal button. This is the end of the axon. In the terminal button, all of these little circles are called synaptic vesicles, and in them are neurotransmitters. When the action potential reaches the neurotransmitter, some of these synaptic vesicles fuse with the membrane right here, and the neurotransmitters are emptied into the synapse. So this space right down here is the synapse. Okay, so this would be the axon and terminal button of one neuron, which we would call the presynaptic neuron. And then this one would be the dendrite, of a postsynaptic neuron. And here there would be a lot of receptors for those neurotransmitters. And so the neurotransmitters would bind to those receptors and have an impact on the postsynaptic neuron. So the process by which this is released is called exocytosis. So again, just kind of to review, these synaptic vesicles, which you see right here, are filled with neurotransmitters, and they're stored near the presynaptic membrane. So this part is the presynaptic neuron. We're in the terminal button. This is where the synapse is. When the action potential reaches the terminal button, these vesicles then fuse with the membrane and release their context, which is neurotransmitter, into the synaptic cleft. So here we're looking at a presynaptic neuron. So there's the axon and terminal buttons. This is the postsynaptic neuron. This is the dendrite. So when the presynaptic neuron releases neurotransmitters, so here you have, these are synaptic vesicles, the red dots are the neurotransmitters. When the action potential reaches the terminal button, the synaptic vesicles have fused with the membrane, and the neurotransmitters are released into the synapse right here. The neurotransmitters then bind with receptors on the postsynaptic neuron. So that's what's happening right here. They then either depolarize or hyperpolarize the postsynaptic neuron. So they change the membrane potential of this neuron, which influences whether this neuron ends up firing an action potential itself or not. So postsynaptic potentials, these are what's occurring in the second neuron, the one that just received the message. It's brief changes in resting membrane potential, and they influence whether or not that neuron will fire its own action potential. When an action potential occurs, we say it fires. There are two types: excitatory postsynaptic potentials, which depolarize the membrane, meaning that it takes it closer to that threshold of excitation. Therefore, they increase the likelihood that a neuron will fire. Then there are IPSPs, or inhibitory postsynaptic potentials, which hyperpolarize the membrane, meaning it takes the resting potential, goes further from the threshold of excitation, and that decreases the likelihood that a neuron will fire an action potential. So that's what's happening. So now I want to talk a little more about neurotransmitters. So not all neurotransmitters can bind to any receptor. Neurotransmitters bind to specific receptors. They have to fit in the right receptor. So a molecule of dopamine is not going to bind to a serotonin receptor. It's only going to bind to a dopamine receptor. So receptors are located, we're talking primarily about ones that are located in the postsynaptic membrane. There are multiple receptor subtypes, meaning that dopamine binds to dopamine receptors, but there are multiple types of dopamine receptors. Serotonin binds to serotonin receptors. There are at least 14 types of serotonin receptors. We can broadly divide receptors into two categories: ionotropic and metabotropic. Ionotropic receptors are ligand-activated ion channels. A ligand is just something that binds to a receptor. So we're talking now about neurotransmitters being ligands. So the neurotransmitter binds to the receptor, and then it causes a channel to open or close immediately. So it's more simplistic. A metabotropic receptor involves, again, a ligand binding to a receptor, but that then causes a change inside the cell and the impacts can be more complex, slower, and longer lasting. So this diagram over here is showing an ionotropic receptor on the top. So down here's the receptor. Here's your neurotransmitter. When the neurotransmitter binds to this receptor, it now opens this ion channel right here, and ions can flow in or out of the cell. So it's fairly simplistic. Down here's a metabotropic receptor. It has this, they both have different types of proteins that span the membrane. Here's the receptor, here's the neurotransmitter. When the neurotransmitter binds to the receptor, it causes a change in this protein, which causes a subunit of this protein to break off and have impacts in other parts of the cells, which could include opening an ion channel. So we mainly have talked, and we'll talk more about receptors on the postsynaptic cell, but there are also some receptors on the presynaptic cell, the one that fired the neurotransmitter. These are called autoreceptors. They are on the presynaptic membrane, they're metabotropic, and they bind to a neuron's own neurotransmitter. So if this is a serotonin neuron, then this is going to bind to serotonin. And basically, they serve to monitor the amount of neurotransmitter in the synapse and help to regulate the release, so that if there's not very much, they might increase release. If there's a lot, they might decrease release. So once a neurotransmitter is released into the synapse, it doesn't stay in the synapse forever. It doesn't stay bound to a receptor forever. There are two primary ways that neurotransmitters are deactivated. So one way, so here again we have a terminal button, here's a synaptic vesicle, here's a neurotransmitter. They were released into the synapse, they bound to a receptor on the postsynaptic membrane. Now what can happen is that they're taken back up into the presynaptic membrane and can be recycled. So this is called reuptake. And so some drugs work by blocking this process. SSRIs, selective serotonin reuptake inhibitors, this includes like Prozac and Zoloft, work by blocking the reuptake of serotonin. Therefore, you have more serotonin staying in the receptor for longer. The other mechanism is for neurotransmitters to be destroyed by deactivating enzymes. So again, the neurotransmitter is released into the synapse, it can bind to a receptor, but then in this case you have these deactivating enzymes that are breaking down the neurotransmitter so that it's no longer the same thing, it's not activated as that. We see that with acetylcholine and acetylcholinesterase. So acetylcholine is a neurotransmitter, acetylcholinesterase in the synapse breaks down acetylcholine into acetate and choline, so it can no longer bind to acetylcholine receptors. And so some drugs work by impacting that process. So, why are we talking about all this in a course about psychoactive drugs and behavior? Coming back to neurotransmitters now. These are chemicals that are going to transmit information between neurons, and they're involved in every aspect of human behavior. Specific neurotransmitters bind to specific receptors. Again, dopamine binds to dopamine receptors. Norepinephrine binds to norepinephrine receptors. This is an important concept here now, how drugs can affect. They can be excitatory, meaning they can increase the likelihood a neuron will fire, or inhibitory, meaning they decrease the likelihood a neuron will fire, or they can be both, and it may depend on what receptor they are binding to. This is extremely important now. You really need to learn and understand this, because this is kind of a basic tenet of understanding how drugs influence behavior. Drugs can be agonists or antagonists for a neurotransmitter. If it's an agonist, it increases the efficacy of the neurotransmitter, so it somehow increases the impact of the neurotransmitter. If it's an antagonist, it decreases the efficacy of a neurotransmitter. We're going to get to some examples of this in a minute. But again, very important concept. So, let's look at what happens in the life of a neurotransmitter, okay? First, neurotransmitters have to be synthesized from precursors. So here we're seeing a neurotransmitter being put together to be synthesized. Then they're going to be stored in synaptic vesicles. Some neurotransmitters might leak from their vesicle and be destroyed by degrading enzymes in the terminal button. But when they're in the synaptic vesicle, when the action potential reaches the terminal button, the synaptic vesicle fuses with the presynaptic membrane, right? The neurotransmitter is then released into the synapse. It binds to a postsynaptic receptor, and then it's either deactivated by experiencing reuptake here, or it's degraded by enzymes in the synapse. And then we also talked about how neurotransmitters could bind with a receptor on their presynaptic membrane called an autoreceptor, which can influence subsequent release. So that's what happens in neurotransmitter action. So now let's look at drugs that are agonists versus antagonists. So drugs that are agonists are going to increase the effect of a neurotransmitter. So they could do this by increasing the synthesis of the neurotransmitter, so that could be their precursor to the neurotransmitter. They could increase the number of neurotransmitter molecules. If they destroy the things that destroy the neurotransmitters, fewer neurotransmitter molecules will be destroyed and you'll have more neurotransmitters. They could increase the release of the neurotransmitter from the terminal button. They could bind to an autoreceptor and block it so that it doesn't then impact future release. So it doesn't decrease future release by binding to this receptor instead of the neurotransmitter. They could bind to the postsynaptic receptor and activate it like the neurotransmitter normally would. Or they could block deactivation. So they could block the reuptake, like the selective serotonin reuptake inhibitors do, or they could interfere with the deactivating enzymes. So all of those things would increase the effect of a neurotransmitter. Antagonist drugs are going to decrease the effect of the neurotransmitter. They could do this by blocking the synthesis of the neurotransmitter. They could do this by causing them to leak from vesicles or degrading them more, the neurotransmitters more. They could block the release from the terminal button. They could bind to autoreceptors and activate them to inhibit future release. They could bind to a postsynaptic receptor and not activate it, so they're blocking another neurotransmitter from binding there. So agonists increase the release and the effects, antagonists decrease the effects. So let's now look at the most important types of neurotransmitters. GABA is the most abundant inhibitory neurotransmitter. So it's going to primarily be involved with decreasing activity. It's involved in anxiety. So most anxiety drugs are GABA agonists. So Xanax, for example, is a GABA agonist. It increases the effect of GABA. Sleep, drugs that are GABA agonists are going to increase sleepiness. And seizures, drugs that are used to treat epilepsy or seizures are frequently GABA agonists to reduce brain activity. So some GABA agonists are benzodiazepines. This includes things like Xanax, Valium. Barbiturates, which are not used very much anymore because they have a high overdose potential, but this includes things like phenobarbital. They are still used sometimes to treat epilepsy, but they're no longer used to treat anxiety. Alcohol is a type of GABA agonist as well. Anticonvulsants, so these are anti-epilepsy drugs, are frequently GABA agonists. Glutamate, on the other hand, is the most abundant excitatory neurotransmitter. So it's usually involved in increased levels of firing. It's very important for learning and memory. So we see that when people are animal models learn new things, you have a change in kind of the structure of the synapse that's mediated by glutamate. It can also be involved in excitotoxicity. This means that high levels of, really, really high levels of glutamate release can actually kill neurons. This happens in strokes, for example. Glutamate, there are a couple of different types of glutamate receptors. This is just showing you one type of glutamate receptor and an MDA receptor. You have a glutamate binding site. You also have other chemicals that bind. And in this case, it controls a calcium channel. In order for this to work, there's this magnesium ion that has to be removed from the channel, which happens if the membrane is depolarized, as well as glutamate has to be bound to open this channel. So PCP, which is a unique drug, is actually a glutamate antagonist. And so one of the things it does is inhibit learning and memory. Ketamine and alcohol are both also glutamate antagonists. Norepinephrine we talked about briefly before. It's involved in activity of the sympathetic nervous system, so it's going to have effects like increasing heart rate and blood pressure. It's involved in stress, again with activity of the sympathetic nervous system, but it's also involved in helping you to pay attention. It can be involved in mood regulation. Some antidepressants work by being norepinephrine agonists. So some norepinephrine agonists: cocaine, And so you'll see that cocaine, for example, stimulates the sympathetic nervous system. Amphetamines, including some medications to treat ADHD, although they have a large impact on dopamine as well. MDMA, or ecstasy, is a norepinephrine agonist, and some antidepressants are also norepinephrine agonists. Blood pressure medications to decrease blood pressure are usually norepinephrine antagonists. Dopamine is involved in a wide variety of things, so smooth motor control. Again, degeneration of some dopamine-releasing neurons is Parkinson's disease, which is why we have Michael J. Fox here. It's involved in the reward pathway, which I will get to in more detail in a little bit. It's also involved in schizophrenia. High levels of dopamine activity in certain regions of the brain seem to be what causes schizophrenia symptoms. So some dopamine agonists, L-DOPA, levodopa is actually the precursor to dopamine. This is what's given to people with Parkinson's disease because dopamine itself cannot cross the blood-brain barrier. So if you give someone with Parkinson's disease dopamine, it's not going to have any impact because it's not going to get through the brain. L-DOPA, however, can cross the blood-brain barrier and then be converted into dopamine. Cocaine is a dopamine agonist. Amphetamines are dopamine agonists, including those that are used to treat ADHD. And then antipsychotic or anti-schizophrenia medications are frequently dopamine antagonists, so they block the effects of dopamine. Serotonin, which is frequently abbreviated 5-HT, involved in a wide variety of things, including regulation of sleep, mood regulation, so a lot of antidepressants, if not all, influence serotonin, eating, hunger and eating behaviors, pain regulation, including migraines, impulse control. So low serotonin is associated with being more impulsive. So most antidepressants, if not all, are serotonin agonists. Some of them, I said, work by blocking the reuptake of serotonin. MDMA, or ecstasy, is a serotonin agonist. Pretty much all drugs that cause hallucinations are serotonin agonists, and they work at a specific type, the serotonin 2A receptor. Cocaine is a serotonin agonist. There are some atypical antipsychotics used to treat schizophrenia that are serotonin antagonists. Acetylcholine is involved in muscle contractions, which is why we've got this guy with this huge bicep. Also the parasympathetic nervous system, as we said before, as well as learning and memory. We see that in Alzheimer's, actually, people seem to have lost a lot of acetylcholine neurons in certain areas of the brain. It can also be involved in the sleep-wake cycle. There's actually two types of acetylcholine receptors, muscarinic receptors, and these are generally involved in parasympathetic nervous system activity, as well as nicotinic receptors, which occur at the neuromuscular junction. And so that's where a neuron meets a muscle. And so the neuron releases a sort of acetylcholine, which binds to the muscle fiber, which causes contraction. An agonist of acetylcholine, as you may have guessed, is nicotine, which is why this is named nicotinic receptor. Antagonists, atropine, which can be used to paralyze muscles during surgery, curare, which causes paralysis, and Botox. Botox works by causing paralysis of muscles, so it works by inhibiting acetylcholine release. Endorphins, you guys have probably heard of. These are endogenous opioids. A common one is beta endorphin. These are involved in analgesia, so reduction of pain. They're involved in feelings of reward. They're thought to be implicated in the runner's high experience. So opioids are agonists here. Opioids we talked about briefly include heroin, morphine, Vicodin, Oxycontin, oxycodone, codeine, things like that. Those are all agonists for endorphins. Naloxone is an antagonist. It's used largely in research, but it's also what you give people if they've had an opioid overdose to try to save them. Endocannabinoids, so we actually have chemicals in our body that are similar to marijuana. We don't have actual THC in our body, but anandamide is an example of an endocannabinoid. They're involved in a wide variety of things, including analgesia, sedation, hunger, bronchodilation, which is the dilation of your bronchi, your bronchial passages for lungs, which is why marijuana can actually be used to help with asthma. THC, one of the active ingredients in marijuana, is an agonist, as is acetaminophen, the active ingredient in Tylenol. They actually did a study where they used an endocannabinoid antagonist, and they gave it to people, and then Tylenol no longer reduced their pain. So I want to go briefly back to the question of why do people use drugs, and get in and give you a neuroscience answer of it, going to like some of the kind of larger drugs and brain questions. Some of this information was in chapter 17, but now that you have more of a brain background, I want to get into it in more detail. So the easiest answer to that question is because it's reinforcing. People use drugs because they get some sort of pleasure out of it. The drugs that are most addictive tend to have the most immediate effects, meaning that people feel the high from them very quickly. So sudden reinforcement is more potent and more addictive than gradual reinforcement. So heroin and morphine are basically the same, except for that heroin crosses the blood-brain barrier more easily than morphine, and so a rush of the molecule then gets into the brain very quickly. On a neuroscience level, reinforcement seems to be caused by the release of dopamine in the nucleus accumbens. So this is caused by neurons that project from the ventral tegmental area, which is a midbrain structure, and project to the nucleus accumbens, which is part of the limbic system. And so when these neurons release dopamine into the nucleus accumbens, people or animals experience feelings of reward. It's a positive reinforcement. All drugs that have an addictive potential cause release of dopamine in the nucleus accumbens because they cause activation of this reward pathway. Natural reinforcers also cause activation of this pathway. This is like food, sex, water when you're thirsty, human contact, mothers looking at their infants cause the release of dopamine in the nucleus accumbens. Again, drugs of abuse cause this as well as other addictive behaviors like gambling. Okay, so if we look, here's a diagram of the brain. And what I want you to look at here primarily is the red pathway. So these are showing different dopamine pathways. But here's the ventral tegmental area, which is in the midbrain. And that then projects to the nucleus accumbens right here, which is part of the limbic system. So it's the release of dopamine along this pathway that seems to cause feelings of reward. But you'll see that there's also some input into the amygdala, which is involved in emotion, and some frontal lobe pathways as well. What we find is that if you give a dopamine antagonist, so if you block the effects of dopamine in this pathway, animals will no longer self-administer drugs that they would normally administer. So you block the rewarding effect here. So we're very confident that this is basically the reward pathway. People also do drugs for negative reinforcement. Negative reinforcement is taking away something aversive. So drugs tend to eliminate negative feelings or, as we talked about before, can decrease withdrawal symptoms. So why are some people more vulnerable to drug abuse than others? We talked about some psychological and social risk factors. Now getting back to some biological risk factors. One seems to be deficient dopamine activity. They did a study where they gave drug-free men, so these were men who didn't abuse drugs, methylphenidate. This is actually Ritalin. It's a dopamine agonist. And they asked them how they felt. Some people kind of felt neutral, some didn't like it, some did like it. The people who reported feeling pleasure had the fewest dopamine receptors in the group. So they had fewer dopamine receptors than the people who didn't really like it or felt neutral about it. And so it's thought then that people who naturally seem to have fewer dopamine receptors may be less able to feel levels of pleasure without drugs. And so drug use is a way to try to kind of normalize their dopamine activity. We see that a number of drug users have reduced activity in parts of the prefrontal cortex here. So the prefrontal cortex is involved in planning and impulsive behaviors, and so decreasing that activity may decrease their ability to make kind of the best decision to not do drugs that have a harmful impact on their life. We also see some relation with the hippocampus. So the hippocampus is involved in learning and memory. So it's involved in associative learning, which is learning that the drug seems to cause the pleasurable effects, so then you want to do it in the future. It seems to also, through its connections with the frontal lobe, be involved in enhancing cravings for some drugs. So that is it for this chapter.